Hi there, welcome to the third and final vodcast on the nervous system. In this one, we're going to spend some time talking about synapses, which is, as we've mentioned already, the space between adjoining neurons. So how the signal gets from one neuron to the all right. So we know that the action potential, as it works its way down, the axon gets to the end at the output zone. And from there, that signal actually promotes the release of a neurotransmitter out into the space at the end of the neuron. And that neurotransmitter, there's a number of them out there. And in essence, these act like hormones. Well, they don't act, they are hormones, to cause a signal to the next neuron itself. So it actually diffuses across the cleft, which is that space, into the input zone of the next cell. Okay, and whether that is a muscle cell, whether that is another neuron, it's a bunch of different options for what that's going to be. Some of the examples of these are acetylcholine, serotonin, dopamine, GABA. These are all types of neurotransmitters that cause a bunch of different effects. Okay. And what we have, the first cell, the presynaptic cell, this is what's going to store the neurotransmitter into a synaptic vesicle. So literally wrapped around what would be like a cell membrane, bilipid layer. So it basically packages it to be pushed out. And you can actually see that, happening right here. Okay, here's those vesicles right there that are going to then join and literally force this neurotransmitter out into the space, okay, which is right in here as well. And when it does this and it stores it, it has gated calcium channels at the end of the axon. And these gated channels are going to open, okay, to allow calcium to move, which will then cause the neurotransmitter to be released. Okay, so those channels open, calcium rushes into the neuron, and voila, the vesicles fill with the neurotransmitter, fuse with the membrane, and release it. So those gated channels are there primarily to promote the release of the neurotransmitter out into the synapse. At the postsynaptic cell, okay, we have the neurotransmitter is going to bind with a protein receptor. Remember, we mentioned that all hormones, by definition, are chemicals that bind at a receptor to elicit a response, and all receptors are proteins. So the same thing goes for these guys. So binding with this is going to alter the receptor and allow ions to diffuse in, which could then, and usually do, initiate another signal, another action potential down the adjacent neuron. Okay, so we have a few types of responses that are going to happen. You can have an excitatory or an inhibitory response, just as it sounds. Something's either going to cause something to happen or it's going to prevent something from happening. Pretty straightforward. So an excitatory neurotransmitter is going to drive that next cell into another another action potential and actually fire it. Something like acetylcholine will do that. Okay, and we'll learn a little bit more about this down the road. An inhibitory neurotransmitter will take it away from an action potential and actually cause it to calm down and not send a signal. Okay, GABA is an example of this. And then we have a group of types of cells, neuromodulators, that are going to either enhance or they might reduce the effects of the neurotransmitter. Okay, some of these chemicals are substance P, endorphins. These kinds of things are going to actually either promote or actually pull down the effect of a neurotransmitter on its adjacent, on whatever cell it's acting at. All right. So integration, how we actually move the signal along. Okay, an EPSP versus an IPSP. So lots of anagrams here for you. Okay, so here's what we have. An action potential, we know, would look like this spike. We've learned about it. We know what it looks like. Okay, so you're going to have certain types of signals that are going to drive closer to the threshold to elicit the action potential. You're going to have inhibitory signals that take it away. They're preventing that threshold from being reached. So lots of neurons, and lots of inputs, are coming into these neurons all at one time. So how do we control those signals so that the thing, basically, in essence, the neurons, its eyes aren't crossing over the number of signals it's receiving? How does it know what to take, what to ignore, and things like that. So it has to balance out these signals and have an effect on that, or what we call an integrated effect. So in some cases, you're going to get this excitatory signal coming in, pushing it to threshold. You're going to have inhibitory signals coming, preventing it, and you get this sort of in-between average, if you will. That is what we call the integrated potential. Okay, so is it going to be enough to go towards action potential or enough to pull it away? So removal of the neurotransmitter. Diffusion away from the cleft. We're going to actually pull the neurotransmitter out, and it's going to be reuptake, going to pull it back in to the actual neuron. So that could happen. It could also be broken down by enzymes. Okay, many of our neurotransmitters are amino acid derivatives, so they're amine, part of the amine group, and so we could break those down. Or transport proteins, literally will take them and recycle them. Okay, so that would mean, for example, in the presynaptic neuron, you've got these proteins that literally grab that neurotransmitter, pull it back into that presynaptic neuron to be used again. The information flow. How do we move information from the point of when we get the signal in and we get the response and the stimulus to actually doing something about it? Okay, and basically we are wired almost like a circuit board to do this, and pretty kind of neat stuff. So our nerves, what we have are our nerves, which are bundled groups of the dendrites of tons, okay, of sensory neurons, and the axons of motor neurons. They all get bundled together, and this is what we refer to as our peripheral nervous system, because they're going to take in all of the stimulus that we have. They extend from our central nervous system, which includes our brain and our spinal cord. Okay, so from there, so peripheral, right, to the outsides. So our central nervous system, and then we've got our peripheral, and that's going to be all the signals will come in, channel it through the brain and the spinal cord. Okay, all of these bundles are bundled up and packaged in a way with connective tissue. Okay, so remembering our tissues we talked about earlier on, so connective tissue is going to hold them together and maintain them so that they stay together, basically. And as we know, our myelin sheaths of our axons help to speed up this transmission, and it creates that insulation effect, which is going to, as the theory goes, pool all of those sodium ions into the nodes so that we can get quicker response and quicker action potentials down them. And these myelin sheaths are created by Schwann cells that secrete the myelin. And that's all a Schwann cell is. It's what we call a neuroglial cell, so it's an accessory to our nerve cells, part of the nervous system, as sort of the helper outer, if you will. Okay, so what they do is they create these wraps around there so that the reactions and the action potentials only happen in the exposed part of the axon. And that's when we get this thing called saltatory conduction. Okay, they only need to be completely open at the nodes between the sheets. Okay, so any channels that are in this area here, forget about it, who cares? Okay, they don't need to be open because they're wrapped up. We only care about these ones that are in the node in the opened area, and that is what's going to jump our action potential from here, okay, and down, okay, so that we can create this one-way street movement down the axon. Some of the problems that can arise with the myelin sheaths, there are some disorders that can come up in some diseases. One in particular is multiple sclerosis, or MS, which I'm sure you've heard about. And this is when it's basically an autoimmune disease, and your immune system kind of misinterprets what the myelin sheaths are and what your Schwann cells are, and it thinks that it's foreign, and so your body begins to degrade the myelin. It basically attacks it and breaks it down, thinking that it's not supposed to be there. So of course in that case, if you now have axons that aren't myelated, signaling doesn't happen as rapidly, and there's a loss of control that can occur, and this is unfortunate and can lead to a lot of lack of muscle control as the signals to all of our muscles, and that includes smooth muscle, that includes skeletal muscle, cardiac muscle, okay, all these things that don't end up getting the signals that they need to. And lastly, reflex arch. This is where we're going to end today. This is how we get simple movements in response to a stimuli. So here's just an example. Okay, how do I know when I'm picking something up? You know, think about it. Ever pick something up that your brain, you know, considers to be heavier than what it is, right? You go to pick it up and you're like, whoo, you know, you could like throw it over your head, right? Because you engaged more muscles. So there's a certain type of stimulus and stretch response that occurs, and you guys can see this. This is right in your book in chapter 34, because that's where we still are, chapter 34, woo, okay? And this reflex arch, so if you go into there, you can see this diagram as well, if this is hard to see. So you've got your bowl of fruit, right? Put it in, okay? Muscles are holding it, right, because we get a stretch response. What happens is that stretch response sends a signal, okay, all the way up to your brain, okay? All these sensory neurons are being sent up, okay, and these sensors, action potentials, boom, 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 okay? Once it hits the brain and the spinal cord, okay, the axon endings then send out a signal via neurotransmitter into the synaptic cleft, okay, to stimulate your motor neuron, and it sends it back. Because remember, signaling is all in a one-way street. So if I take a neuron up to my central nervous system, it's going to take a different neuron to go back to my muscle. So as soon as I get back down, axon endings are going to attract to the actual muscle fibers, to the muscle cells, releasing acetylcholine into the nerve, okay, into the muscle, you know, from the nerve into the muscle fibers, and that stretch response allows us to keep the bowl steady. Okay, because we're maintaining a contraction of our bicep muscle. Okay, and we feel the stretch of the pull down is what's initiating the signal this way to provide the amount of muscle fibers that we're going to need to engage to make sure that we keep something upright. Okay, so something like this, right? I know based on the stretch response coming down initially, so in here, that this isn't really that heavy and I don't need to engage as many muscle cells. And that's how it works. So it's a pretty neat little thing in terms of what we call a reflex arch. So with that, we are going to end our look at the nervous system and not spend a whole lot of time on it, but I just wanted to give a little bit of insight into how signals get sent. So that's where we're going to end it. From here, we're going to begin to look at the cardiorespiratory. We're going to start to look at the cardiorespiratory systems and look at how they work to maintain homeostasis in humans and as well as other organisms too. So have a great night, and we'll catch you guys later. Take it easy.